Hey everyone, and welcome back for another deep dive.
It's great to be here.
Today we're going super small to explore the world of nucleic acids,
You mean?
DNA,
Oh, okay, got it.
and RNA.
The blueprints of life.
Exactly, the blueprints and the builders of life itself.
That's a good way to put it.
We've got some lecture notes and some diagrams to help us break down how these tiny molecules,
Tiny.
yes, tiny molecules, hold the keys to heredity.
It's amazing, isn't it?
It really is, and by the time we're done with this deep dive, you'll have a much better grasp of molecular biology's core concepts.
Yeah, they're really not as complicated as some people think.
No, they're really not. And once you get them, it's really fascinating.
It is.
So let's start with the basics.
Okay.
We hear a lot about DNA, RNA, and proteins.
All the time.
But how are they all connected?
Okay, well, think of it this way. DNA is like a master instruction manual. It's locked away in a safe place inside the cell's nucleus.
Okay.
RNA is the messenger that transcribes those instructions and carries them out to the construction site where the proteins are built.
So the proteins are the builders.
Exactly. And this whole process from DNA to RNA to protein is what's called the central dogma of molecular biology.
Okay, so the central dogma,
Yeah.
DNA holds the instructions, RNA is the messenger, and proteins are the builders.
You got it.
Okay, but what are these molecules actually made of?
They're all built from these tiny building blocks called nucleotides.
Nucleotides.
Yeah, imagine them like the letters of the genetic alphabet.
Oh, so just like letters form words, nucleotides string together to create the language of life.
That's a great way to think about it.
That's cool.
Yeah.
So tell me more about these nucleotides.
Okay.
What makes them up?
Okay, so each nucleotide has three main parts.
Okay.
A sugar molecule,
Okay.
a phosphate group,
Uh-huh.
and a nitrogenous base.
Right.
Now, the sugar is slightly different in DNA and RNA.
Okay.
It's deoxyribose for DNA
Okay.
and ribose for RNA.
Got it.
But it's the nitrogenous base that really steals the show.
Okay. The nitrogenous base.
Yeah.
Why is it so important?
Because there are four main types of these bases in DNA.
Okay.
Adenine, guanine, cytosine, and thymine.
Wow, that's a mouthful.
I know, right?
Okay.
We usually just abbreviate them as A, G, C, and T.
Oh, thank goodness.
And RNA uses the same bases, except it swaps out thymine for uracil.
Which is you.
Yes.
Okay.
And here's the key. The specific order of these bases, like a sequence of letters, encodes the instructions for building proteins.
So the sequence of these bases is like a code that determines what proteins get built.
Exactly.
That's pretty amazing.
It is.
But we've been talking about single strands, and I know DNA is famous for its double helix structure.
Oh, yeah, absolutely.
Right.
And that structure is key to DNA's function.
Okay.
It's like a twisted ladder
Uh-huh.
with the sugar-phosphate backbones forming the sides
Okay.
and the nitrogenous bases pairing up in the middle to form the rungs.
So it's not just a single strand.
No.
How are the two strands held together then?
They're connected through those nitrogenous bases.
Okay.
And the pairing isn't random.
Okay.
A always pairs with T,
Uh,
and G always pairs with C.
interesting.
And these pairs are held together by hydrogen bonds,
Okay.
which keep the two strands tightly connected.
So the double helix is stabilized by these base pairs.
Exact.
It sounds like a pretty ingenious design.
It really is. This structure is what allows DNA to replicate itself accurately,
Okay.
ensuring that the genetic information is passed down faithfully from one generation to the next.
Wow. So this double helix structure is responsible for inheritance itself.
It is.
That's a big responsibility for something so tiny.
I know, right?
But speaking of replication, how does DNA actually copy itself?
It's a pretty intricate process.
It seems like it would be.
It is, but it's called semi-conservative replication.
Semi-conservative replication.
Yeah, and it's one of the most elegant processes in molecular biology.
Okay.
Essentially, the two strands of the double helix unwind and
Yeah.
and each strand serves as a template for a new complementary strand.
So each new DNA molecule ends up with one original strand and one newly synthesized strand.
Exactly.
Clever. But unwinding that entire helix and making sure everything gets copied correctly, it must be a very intricate process.
It is, and there are a lot of molecular players involved.
Oh, I bet.
Yeah, for instance, there's an enzyme called helicase that unwinds the helix,
Okay.
and another one called DNA polymerase that adds the new nucleotides to the growing strand.
So there's a whole team of enzymes working together to make sure replication happens smoothly.
That's exactly right.
That's amazing. But before we get into all those details, I want to take a step back in time for a minute.
Okay.
And talk about how scientists actually figured out that DNA was the carrier of genetic information.
Yeah.
Because, I mean, there was a time when they weren't even sure it was DNA.
Right, they thought proteins might hold the key.
Really? It's hard to imagine a time when we didn't know that DNA was so central to life.
No.
So what led scientists to that discovery?
Well, in the early 20th century, scientists knew that chromosomes carried genetic information, but they weren't sure which molecule, DNA, or protein was responsible.
So how did they finally crack the code and figure out that DNA was the star of the show?
Well, it took a series of brilliant experiments.
Okay.
One of the first was Griffith's experiment.
Griffith's experiment.
Yeah, which showed that a transforming principle
Transforming principle.
from heat-killed virulent bacteria could turn harmless bacteria into a deadly form.
So something from the dead bacteria was transforming the live ones.
That's right.
Sounds like a spooky sci-fi movie.
It does a little bit, right? But it was a groundbreaking discovery.
Okay.
And then later, the Hershey-Chase experiment solidified DNA's role.
Okay.
They used viruses called bacteriophages.
Bacteriophages.
Yeah, they infect bacteria.
Okay.
And so they labeled the protein coat of some viruses
Wow.
and the DNA of others with radioactive markers.
Oh, right.
And then they let them infect bacteria.
So by tracking the radioactivity, they could see what part of the virus actually entered the bacteria.
Exactly. And the results were clear. Only the radioactive DNA entered the bacterial cells,
Wow.
proving that DNA was the genetic material injected by the viruses.
That is amazing. Such an elegant way to prove DNA's role.
It is a beautiful experiment.
Yeah. But knowing DNA was important was just the first step, right?
Right.
They still needed to figure out its structure
Yeah.
in order to understand how it actually worked.
Exactly. And that's where Chargaff's rules come in.
Chargaff's rules.
He analyzed the base composition of DNA from different organisms,
Okay.
and he found that the amount of adenine always equaled the amount of thymine,
Okay.
and the amount of guanine always equaled the amount of cytosine.
So he noticed these specific ratios of the bases.
Yeah.
Did that give him any clues about the structure?
It did. These consistent ratios hinted at the idea of base pairing, like puzzle pieces fitting together.
Okay.
But the real breakthrough came with Rosalind Franklin's famous X-ray diffraction images, which revealed the helical structure of DNA.
Her work was essential for cracking the code, wasn't it?
It really was, absolutely.
Yeah.
Her images provided crucial data for James Watson and Francis Crick to build their groundbreaking model of the double helix,
Right.
which incorporated the base pairing and explained how DNA could replicate.
So they put together all the pieces of the puzzle.
They did.
Chargaff's ratios,
Oh.
Franklin's images, and their own insights to create this now iconic double helix model.
Exactly. They figured out that the two strands of DNA run in opposite directions,
Okay.
with the bases paired up in the middle like rungs on a ladder.
Mm-hmm.
And this structure elegantly explained how DNA could replicate itself, with each strand serving as a template for a new complementary strand.
It's fascinating how scientific discoveries often build upon each other like that.
It really is.
Each researcher adding another piece to the puzzle.
Absolutely. And this discovery was a turning point in our understanding of life. But understanding the structure was just the beginning. The next question was, how does this replication actually happen?
Right, because knowing the structure is one thing, but actually seeing it in action
Yeah,
must be a whole other level of complexity.
it is. And that's what we'll talk about next time.
Can't wait.
Me neither. This is fascinating stuff.
It is.
So we've got this elegant double helix structure, right?
Yeah.
But how does it actually unwind and copy itself?
Yeah, it seems like it would be really complicated.
It's a bit like a carefully choreographed dance
I like that, a molecular dance.
with lots of molecular players involved.
Tell me more about how this all unfolds.
Well, it all starts at specific points on the DNA molecule
Okay.
called origins of replication.
Orders of replication.
Imagine these as tiny zippers that start to unzip the DNA double helix.
So the DNA starts to unzip at these specific spots.
Right.
What happens next?
As the DNA unwinds,
Yeah.
it creates these bubble-like structures
Yeah.
called replication forks.
Replication forks.
At each fork,
Okay.
there's a leading strand, which is synthesized continuously in one direction,
Okay.
and a lagging strand, which is synthesized in short pieces called Okazaki fragments.
Okay, so why is there a difference to the leading and lagging strands?
It all comes down to the direction in which DNA polymerase can work.
Okay.
Remember, that's the enzyme that builds the new strands.
Right.
It can only add nucleotides in one direction.
Okay.
So the lagging strand has to be synthesized in these discontinuous fragments.
Huh. That's interesting.
It's like trying to lay bricks in one direction.
Okay.
You can lay them down smoothly in one direction, but if you try to go the other way, you have to work in sections.
That's a great analogy.
So the leading strand is laid down smoothly, while the lagging strand is built in these little chunks.
So what happens to those fragments later?
Another enzyme called ligase comes in
Okay.
and seals up the gaps between the Okazaki fragments.
So it creates a continuous strand.
Exactly. It's like a molecular glue that joins everything together.
Wow. It's amazing how all these different enzymes work together with such precision.
It really is a remarkable process.
But I imagine that replicating such a long, complex molecule
Yeah.
must come with some challenges, right?
You're absolutely right. One of the biggest challenges is replicating the very ends of linear chromosomes.
Why is that a problem?
Well, remember that DNA polymerase needs a primer.
Uh-huh. A short piece of RNA?
Right, to start building the new strand.
Right.
At the very end of the lagging strand, there's no place to lay down a primer for the last fragment.
So it's like running out of track at the end of the line.
Exactly.
The replication machinery can't quite reach the very end.
Right.
So what happens then?
That's where telomeres come in.
Telomeres.
They're these repetitive DNA sequences
Okay.
found at the ends of chromosomes,
Yeah.
acting like protective cap.
Protective caps, what do they protect against?
They prevent the loss of important genetic information during replication
Okay.
without telomeres. The ends of chromosomes would get shorter and shorter with each round of replication,
Oh, wow.
eventually leading to the loss of essential genes.
So they're really important.
They are incredibly important.
So they act as a buffer zone,
Yeah.
making sure that the important genetic information doesn't get chipped away during replication.
Think of telomeres like the plastic tips on your shoelaces.
Oh, that's a great way to visualize it.
They prevent the laces from fraying and unraveling.
I've heard that telomeres are connected to aging. Is that true?
Yes, there is a strong link between telomeres and aging.
Okay.
In most of our cells,
Uh-huh.
telomeres shorten with each cell division.
Okay.
As we age,
Yeah.
our telomeres get shorter and shorter,
Yeah.
and this contributes to the aging process.
So as those telomeres wear down,
Yeah,
our cells become less efficient at replicating.
right.
And this contributes to the wear and tear we experience as we get older.
That's the idea. It's incredible to think that something so tiny can play such a big role in the aging process.
It really is.
And there's this fascinating enzyme called telomerase
Telomerase.
that can actually lengthen telomeres.
Wow, really?
But it's only active in certain types of cells, like stem cells and cancer cells.
So cancer cells have found a way to keep their telomeres long,
Yes,
which helps them keep dividing uncontrollably.
exactly. It's a bit of a double-edged sword, isn't it?
It is. So telomerase is a key player in the immortality of cancer cells.
Yes.
But it's also essential for the renewal of tissues and organs.
Right. Scientists are studying telomerase very closely, hoping to understand its role in aging and disease.
It sounds like there's still a lot we need to learn about telomeres and their role in aging and disease.
There is. We're just scratching the surface.
But even with all of these protective mechanisms in place, DNA replication isn't always perfect, right?
That's true.
I mean, we're talking about billions of base pairs being copied.
It's a lot of information to copy.
So mistakes can happen.
Sometimes a base might get inserted or deleted, or the wrong base might get paired up.
And these changes in the DNA sequence are what we call mutations.
Precisely.
And these mutations can have pretty big consequences, right?
Most mutations are neutral, having no effect on the organism.
Okay.
Some can be harmful, leading to diseases or disorders.
Right.
But occasionally a mutation can be beneficial,
Yeah.
providing an advantage that helps an organism survive and reproduce.
So mutations are the fuel for evolution.
Exactly. Those tiny changes in DNA passed down through generations can lead to the incredible diversity of life we see today.
It's incredible to think about.
It's a reminder that change, even at the molecular level, is a driving force in the evolution of life.
So we've talked about how DNA makes copies of itself, but how does that information actually get used to build something?
Right. We still need to talk about transcription and translation.
Yes, how DNA's instructions are turned into proteins.
That's what we'll cover next.
All right, sounds good. So we've covered DNA replication and how those instructions are passed down.
Right.
But how do those instructions get put into action? How do we go from that DNA code to actually building a protein?
That's where transcription comes in.
Transcription.
It's the process of converting a DNA sequence into a messenger RNA molecule.
Messenger RNA.
mRNA.
mRNA.
Think of it like transcribing notes from a textbook.
Okay.
You're creating a portable copy of the information that you need.
So we're making a copy of a gene, a specific section of DNA,
Right.
that holds the code for a protein.
Exactly.
Where does this copying take place?
Right in the cell's nucleus where that DNA blueprint is stored.
Okay.
An enzyme called RNA polymerase
RNA polymerase.
binds to a specific region of DNA called the promoter,
Promoter.
which acts like a start here signal.
So the promoter tells the RNA polymerase where to begin transcribing.
Exactly. It's like a bookmark in the DNA instruction manual.
Okay, cool.
From there, RNA polymerase moves along the DNA strand, unwinding it and adding complementary RNA nucleotides to build the mRNA molecule.
And the mRNA is like a single-stranded version of that DNA sequence, ready to carry the message out to the protein-building machinery.
Precisely once the mRNA molecule is complete,
Okay.
it leaves the nucleus and heads to the ribosomes in the cytoplasm.
And ribosomes are like the protein factories of the cell.
That's a great way to put it. They're these complex molecular machines that read the mRNA sequence and use it to assemble amino acids into proteins. And this process is called translation.
Okay, so translation is where we finally get to see those DNA instructions put into action.
Yes.
How does a ribosome actually read the mRNA code?
The mRNA is read in groups of 3 nucleotides called codons.
Codons.
Each codon specifies a particular amino acid, which is like a building block for a protein.
Okay.
So think of it as a code book where each 3-letter word corresponds to a specific amino acid.
So the ribosome reads the codons on the mRNA and matches them up with the corresponding amino acids.
Exactly.
It's like a cellular assembly line.
It is a very efficient process.
That's so cool.
The ribosome moves along the mRNA codon by codon, adding amino acids to a growing chain.
Okay.
This chain will eventually fold into a specific 3-dimensional shape, which determines the protein's function.
So we go from DNA to mRNA to a chain of amino acids, and then that chain folds up to become a functional protein.
That's the journey.
That's an amazing journey for genetic information to take.
It's mind-blowing.
But are all genes expressed all the time?
That's a great question. And you're right, not all genes are expressed all the time.
Okay.
There are these complex regulatory mechanisms that control which genes are turned on and off in different cells at different times.
So it's like having this massive library of genes.
Yeah.
But only checking out certain books when you need them.
That's a great analogy, and this regulation is crucial for cell specialization and development.
So for example, a skin cell doesn't need to make the same proteins as a brain cell, even though they both have the same DNA.
Exactly. It's like having all the blueprints for an entire city, but only activating the instructions for building a library when you need one.
Okay, so what determines which genes get turned on and off?
A variety of factors can influence gene expression, both internal signals within the cell and external cues from the environment.
So it's this really dynamic interplay between genes and the environment.
That shapes who we are.
It sounds like there's still a lot we're learning about gene regulation.
Absolutely. This is one of the most exciting areas of research in molecular biology.
But it's clear that it plays a crucial role in how our bodies function.
It does, and understanding how genes are regulated and how this regulation can go wrong is essential for developing new therapies for diseases.
And with all of these new technologies like CRISPR, we're now able to edit genes.
I know, it's amazing.
With incredible precision. It's mind-blowing to think that we can potentially rewrite the code of life.
CRISPR has revolutionized genetic research, offering a powerful tool for studying and manipulating DNA.
Right, it holds so much promise for treating genetic diseases and even engineering crops with enhanced traits.
Absolutely.
It seems like there's so much potential, but also potential risks as well.
Yeah.
It's important to consider the ethical implications as we venture further into this new era of gene editing.
I completely agree. The power to alter the genetic code comes with great responsibility.
It really does.
And it's crucial to have thoughtful discussions about how these technologies should be used.
It's a lot to think about, but that's what makes this field so fascinating.
It really is.
As we wrap up this deep dive into the world of DNA and RNA,
It's been a great journey.
it's clear that DNA is much more than just a molecule. It's a story.
A history book of life itself.
And it's a story that's still being written.
Absolutely, with new chapters unfolding all the time.
From the basic structure of DNA to its intricate replication process, from the central dogma to the complexities of gene expression, the world of molecular biology is full of wonder and discovery.
And we've only just scratched the surface.
We hope this deep dive has sparked your curiosity and given you a newfound appreciation for the molecule that makes life possible.
It's been a pleasure exploring this with you.
So keep exploring, keep asking questions, and never stop marveling at the elegance and beauty of the molecular world.
That's great advice.
Thanks for joining us, and we'll see you next time for another deep dive.
Bye, everyone.